Hello, my name is Lindsey Treviño, and I am currently a postdoctoral research associate in the lab of Dr. Cheryl Walker. As summarized in the Endocrine Society's Scientific Statement on Endocrine Disrupting Chemicals, or EDCs, environmental exposures can have adverse effects on human health. Numerous studies have shown that exposure to EDCs during critical windows of development can reprogram the epigenome and increase susceptibility to diseases in adulthood, a process known as developmental reprogramming. A prime example of EDC-induced developmental reprogramming in the male reproductive system is early life exposure to bisphenol A, or BPA, which results in male genital abnormalities, abnormal prostate size, and elevated susceptibility to prostate tumors. Our lab is interested in understanding the molecular mechanisms underlying EDC-mediated epigenomic reprogramming. We were the first to show that neonatal exposure to EDCs could regulate the methyltransferase activity of EZH2 downstream of non-genomic estrogen receptor, or ER, signaling to PI3K-AKT, resulting in reduced H3K27 trimethylation in the developing reproductive tract. Interestingly, BPA activates non-genomic ER signaling in the developing rodent prostate and human prostospheres. Thus, we asked whether the risk for development of prostate lesions modulated by early life EDC exposure could be due to disruption of methyltransferase activity and reprogramming of the epigenome in the developing prostate. To test this hypothesis, we utilized a well-characterized rat model of hormone-induced prostate cancer in which neonatal BPA exposure increases susceptibility to prostate tumor genesis. We found that in the developing prostate, neonatal BPA exposure activates PI3K-AKT signaling and activates the histomethyltransferase MLL1. Although in silico data suggests that MLL1 may be a direct target of PI3K-AKT signaling, the regulation of MLL1 cleavage and activity by post-translational modifications is not well understood, and future studies to examine this regulation are warranted. Activation of MLL1 in the rodent prostate increases H3K4 trimethylation at genes associated with prostate cancer, including the prostatins and kallikreins, as well as several genes in the prostate cancer KEGG pathway. These genes exhibit persistent elevated H3K4 trimethylation, and in the case of prostatins and kallikreins, is associated with increased basal gene expression. These data are particularly interesting in light of the fact that these genes are putative cancer biomarkers, including for prostate cancer. Hence, changes in epigenetic marks on these genes could potentially serve as biomarkers for future studies exploring the contribution of early life EDC exposure to the etiology of prostate cancer. In contrast to the prostatins and kallikreins, reprogrammed KEGG pathway genes exhibited elevated H3K4 trimethylation, but no change in basal gene expression. Rather, this reprogramming resulted in hyperresponsiveness to hormone, which may contribute to increased susceptibility to prostate lesion development. Interestingly, H3K4 trimethyl reprogramming alone was not sufficient to produce an exaggerated response to hormone, since not all of the reprogrammed genes exhibited increased expression upon hormone treatment. Studies are currently underway to identify additional factors that may play a role in this response. In the current study, we identify a new pathway for developmental reprogramming that involves modulation of MLL1 activity by non-genomic signaling. These data contribute to the increasing body of evidence linking EDC-dependent developmental reprogramming and the dysregulation of histomodifiers responsible for the plasticity of the epigenome. Epigenomic plasticity creates a particular vulnerability to environmental exposures, which, if able to disrupt the developing epigenome, can have far-reaching consequences across an exposed individual's lifetime and potentially for subsequent generations. On behalf of Dr. Walker, the co-first author Dr. Huang, and the contributing authors, I would like to thank the Endocrine Society for providing this opportunity to discuss our work, and I would like to thank you for watching.